Good evening and welcome to the Illinois RiverWatch Winter Speaker Series. Tonight, I am pleased to welcome our presenter, Amy Janik. Amy received her undergraduate degree from University of Wisconsin-Madison, where she majored in conservation biology and Spanish. Amy worked as an avian research assistant, environmental educator, and conservation crew leader before earning her master's degree at Western Michigan University. Amy is currently a research specialist at the National Great Rivers Research and Education Center. A couple of years ago, Amy presented on the initial progress on the Odonates of Illinois project, and tonight she will be presenting the final results of this project. With that, I will gladly turn things over to Amy Janik. Thank you for joining us.
In the beginning, we study the ecology of big rivers, the workings of the watersheds that feed them, and the ties to the river communities that use them. And so kind of my role at NGREC is to kind of focus on the wildlife ecology portion of river ecology and wetland ecology. And so yeah, I've been, oops, sorry about that. I've been kind of using, I've been a co-principal investigator on the Odonate, Dragonfly, and Damselfly project, and it's been running since 2021 to 2025. So I'm kind of going to go over that with you guys today. And so again, I'm going to kind of go through the life history and a background of just dragonflies and damselflies because they have lots of exciting things to learn about them. And so some of this might be a repeat for others, but just kind of wanted to give you that history and kind of their conservation needs and why it's important to study them. And then I'm going to bring the focus back to our project that we did, the objectives and the methods, and then our results and suggestions that we came up with out of the project. And so to start, what are odonates? And so the order Odonata is made up of two suborders. We have the Anisoptera, which are dragonflies, and the Zygoptera, which are damselflies. To kind of shorten it, I refer to them often as odonates. And that's what you'll hear throughout the presentation. So odonates are, especially the adults, are really easily recognizable, with dragonflies being fairly robust and they like to perch with their wings open. And damselflies tend to be slender, and a lot of their species perch with their wings closed. And so that's kind of one of the bigger ways to tell the difference between the two suborders. So the defining characteristics of odonates, they have rather large eyes, if you can't tell. They have tiny antennae, they have chewing mouth parts, and they have well-developed wing muscles. They are carnivorous at all stages of their life cycle. There have been over 6,000 species described in the world, and they can occur on every continent except for Antarctica. And they can be found in a variety of freshwater habitats. They range from kind of ephemeral streams to large lakes, huge rivers, anything really that's freshwater. And so odonates are pretty cool because they first appear in the fossil record about 250 million years ago. So although some groups and suborders have long gone extinct, there are many families that we recognize today that were actually present at the start of the Jurassic period. And they appear before the dinosaurs and then long outlast them, which is very interesting. So the odonate life cycle spans across both aquatic and terrestrial ecosystems. They play key roles in both of those environments. Unlike butterflies, odonates have an incomplete metamorphosis, which means that they lack a pupa or a chrysalis stage between their larva and adult. Odonate nymphs actually stay active in the water while they're developing, and then they molt their larval skin, which is very similar to what we see with cicadas. And so I'm going to kind of walk you through each stage of their life cycle, starting with the egg stage. There are a variety of ways that female odonates oviposit or lay eggs. Some species will just directly put them into the water. Some just drop them from the air and hope that they fall into the water. Other species will actually lay them either onto or into aquatic plants with their ovipositor, seen in that picture. It's really interesting. Some females will lay their eggs while still attached to a male, Some will be abandoned by the male, basically, and it's just like the widow skimmer is actually named a widow skimmer because the female gets abandoned during egg laying. And so they have a lot of unique tactics on how they lay eggs. And adult females can lay a clutch of eggs daily. Each clutch could range from about 100 to 1,000 eggs, kind of depending on the species. Same with the development of the eggs. They can hatch in a matter of 5 days, or it could be like 250 days, based on kind of what the conditions of the ecosystem that they're in. So if it's warmer, they might be able to hatch quicker. When it's colder, they usually take a longer time. There's a lot of variation. So the next stage, we have the nymph or the larva stage. This, I think, for most people is kind of the less familiar group. That might be different for RiverWatch folks, as you guys spend a lot of time with macroinvertebrates. But this is kind of a really fun stage because they spend most of their life underwater. The variation in development time is also quite extreme in this stage. Warmer temperatures, they can complete their larval development in about a month. But in some colder, high latitude streams, maybe some ones in mountains, the larva can take up to like 8 years to fully develop. For a lot of our more common species, it can be about 2 years, 1 year, and then some species can even do it in about 6 months, depending on what time of the year the eggs were laid. And so before I move on to the final stage, the adult stage, I kind of wanted to give you a little bit of a background on that body plan of insects and some really cool morphological characteristics that odonates have. So both nymphs and adults have that typical 3-part body plan. You have the head, you have that middle section, which is the thorax, and then the abdomen usually is the longest part. So one of the more fascinating aspects of an odonate nymph head specifically is the labium, or essentially that lower lip of that mouth part. And so depending on the species, the labium can be either flat or scoop-shaped, more like a spoon. And so it's kind of one of their key characteristics that you use to identify when looking under a microscope whether this is depending on which family it's a part of. So a scoop shape will belong to more of the skimmer family, and then like the flat ones, for example, can be more of the darners, which is pretty cool. And that the importance of that labium is that it allows for like a rapid strike when they're hunting for prey. It essentially acts as like a catapult, because it stores up kinetic energy and it releases and then startles and captures prey. In addition, they also have these like needle-like projections at the end of their labium, and those are called movable hooks. And they can extend that and use that to help hold and or stab a prey. And these are just some photos from the beginning that we took in our lab under our digital microscope, just to kind of look at some up-close pictures of some of the mouth parts that I was seeing. And so the length and the shape of this mouth part was really helping me to narrow down that family or genus that it might be a part of. And so you can see a damselfly mouth part on the right. That center kind of hole in that mouth part is really specific to a broad-winged damselfly. So that one was always a super easy one to kind of figure out. And then the basket-tailed dragonfly, has that kind of scoop-shaped mouth part as well. And so these are usually the first parts that I would look at when trying to identify a nymph. And so then moving to another part of the head, I think the most obvious part of an adult head, at least, is a pair of large prominent eyes. The dragonfly eyes are usually very close together and they touch. Damselflies, on the other hand, are more spread apart and wide. Damselflies tend to be prey more often, and so that kind of how I usually remember that is that they're usually on the side of their heads. Vision is probably the primary sense used by odonates, and in most species, the compound eyes can contain up to 30,000 simple eyes, and those each function independently of one another. One of the many reasons odonate adults are so hard to catch in the field is due to their ability to rotate their head 170 degrees. from their normal position, and they can also tilt it upward about 90 degrees. And this means that they lack many blind spots and make it difficult for people like me to try and catch them with a net. And so the next part of the body plan, we have thorax. And the thorax is essentially the same in adults and nymphs, and they serve the same purpose. It's going to be that middle part where the legs and the wings or the wing pads for nymphs are located. And so the wings of adults are constructed with a really complex set of veins, and those veins support an incredibly thin membrane. They're made of a flexible protein that allows the wings to bend and move and easily recover. The four wings operate independently of one another, and that allows for, optimal flight performance and an increased flight agility. The average flight speed of a medium-sized dragonfly can be around 4.5 miles per hour, but like a skimmer that might be in territorial defense over an open pond can fly up to about 9 miles per hour. And so then we have the abdomen, which takes up most of the odonate body in both the nymphs and the adults. So dragonfly nymphs' abdomens can range from kind of stout and robust, similar to a disc shape, or it can be elongate and cylindrical. Damselfly nymphs are very slender, similar to how their adult bodies are, and narrow. They have these prominent gills that resemble leaf-like appendages at the end of their abdomen, which is similar to kind of how a mayfly nymph also has those. But so the nymph abdomen is probably one of the cooler characteristics of odonates. This is where the gills are located for the nymphs to breathe. So for damselflies, the gills are those, like I said, the leaf-like appendages at the end of their body. They're not just a tail, they actually are how they breathe. And for dragonflies, their rectum contains their respiratory system. So they basically breathe through their butt. But because of the strong diaphragm that they have in their abdomen, water can just be expelled very rapidly from the rectum, which will shoot the nymph forward through jet propulsion, basically allowing them to escape from predators. And so while some nymphs may live underwater for a few years, the lifespan of an adult odonate is much shorter. We'll have an adult dragonfly and damselfly can live between like 1 month to a few months. And during that time, they have very focused lives. They focus on feeding, basking, and reproducing. So the diet of an odonate nymph can vary from large aquatic organisms to very small aquatic organisms. The larger, more developed dragonfly nymphs can forage on small fish or even tadpole, whereas smaller dragonfly and damselfly nymphs can eat things like mosquito larvae, biting fly larvae, or even mayfly larvae. They have a tendency to eat each other. Basically, they will eat whatever fits into their mouths. And for that reason, they are, partly very successful predators. They have the ability to camouflage really well in plant roots and detritus. They can swim very quickly through vegetation, and so they're just really efficient hunters, especially in the water. But they're also super efficient in the sky. Adults have a huge prey range. Tiny insects are a big portion of them. But dragonflies and damselflies, they'll eat each other again, but they also like butterflies. And although this is pretty rare, some of the larger dragonfly darners can even hunt young hummingbirds. And again, they are super successful as predators in the air, just as impressive as well. And so odonates have a couple of different ways to stay away from predators. Some female odonates are pretty cryptic and are able to blend in with their surroundings to avoid being eaten. But they also have a tendency to use their speed and agility, and they have a zigzag flight pattern that they'll use to outsmart their predators while being chased. However, the unlucky ones still become prey to predators such as birds, bats, frogs, lizard, and fish sometimes. So there's quite a few things that still will eat them. While it might be difficult, they still can. A tendency for females to get eaten a lot is while they are ovipositing eggs. That's usually one of the more dangerous times for females. And so odonates are a very colorful group of insects. They represent all the colors of the rainbow. And these vibrant colors are not just for aesthetics. They serve specific purposes. So males are commonly the more color ones. Not always, but it is common. And this is kind of suggested because scientists think that it might make it easier when there's big swarms of dragonflies, that they have less chance of confusing females for males, or males for females when they're trying to select mates. And it also helps with their thermal regulation. So odonates living in more sunny wetlands will have brighter colors to reflect solar rays, and that regulates their body temperature. And certain colors are also handy with camouflage and kind of help them blend into their surroundings. But some of them could also be to attract mates and also to warn off any potential predators. And so mating usually takes place near water. Male dragonflies have a tendency to set up territories, and that's kind of their goal, is that they protect that territory to prevent too many males kind of coming into the area, and to decrease any competition for females. And so a lot of the times you'll see a focus of males being at the water bodies, and then females are usually foraging nearby. And so courtship for females varies among odonate families. Damselflies have a tendency to have very elaborate and flashy courtship. That's similar to how birds might do a courtship display, which is kind of interesting. And so there's specific times of the years where adults want to be out, and that is called a flight season. And this is when you're most likely to see a species. And the timing of a flight season is mostly based on, that larval development period and the environmental conditions of the area. And so some species that are in kind of more volatile, unpredictable weather areas, they tend to have a synchronized emergence and a synchronized die-off. Others will continue to emerge and have a flight season just throughout the summer and fall. Many odonate nymphs will probably hibernate in the water over winter, and then they emerge as an adult in that early springtime. However, it is very rare for an adult to hibernate over the winter. And so since odonates span both aquatic and terrestrial habitats, they are very, very vulnerable to threats that impact freshwater ecosystems and landscapes. So some of the challenges dragonflies and damselflies face are habitat loss, pollution, and climate change. Wetlands serve as one of the most economically valuable ecosystems, and they're disappearing rapidly. Climate change, which is already a threat to many insect species, are just going to lead to more extreme drought and will directly impact odonates, and will probably decimate their populations if they don't have the fresh water that they normally need. And so some species are generalists, which means that they can kind of eat and inhabit various areas, which would allow them to adapt to more urban environments, which we've seen of some species already. However, there are a decent amount of specialist species that may only eat a certain type of prey or need a specific plant to lay eggs. And those species are not going to thrive the same way in urban areas. And so it's positive to know that we have some odonate species that can adapt to a more urban habitat, but those species are still going to be sensitive to, water quality, those pollutants such as road salt, heavy metals, insecticides. Those are still things that are going to be dangerous for both nymph and adult populations. And so due to all these environmental threats that we have for odonates, and basically the fact that they're such important players in ecosystems, it was really important to make sure that we kind of understood what the status of odonates were in Illinois. And so it tends to be conservation of invertebrates or insects tend to be neglected, and many species in need are never recognized due to a lack of information and baseline data. And Illinois has a tendency to lack a lot of the distribution and vulnerability information that we need for families of odonates. And so it's kind of hard to know how to protect an order of insects without this knowledge. And so just to kind of show you, this is a heat map from Odonata Central that I took from 2023 or 2022. And it's just kind of a collection of the records that were on this database in Illinois. And these were all collected through community scientists. And that red area is going to be the area where there are over 30 records of a species. But all the purple is less than that, or no color means no data. And so we can see that the Chicagoland area has a pretty good consistent red mark where there's enough records for odonates. But there's still a greater portion of the state that is understudied. And so it was important for us to conduct an in-depth assessment of the conservation status of this diverse taxon group so that at a state level, that way we can kind of mitigate this lack of information and hopefully offer some suggestions for conservation and management of these invertebrates, which are really important. And so with the need to kind of know some of these knowledge gaps, NGRAC was awarded a state wildlife grant by the state of Illinois. And the goal of this grant was to kind of look into odonates. And we had four main objectives. We wanted to assemble historical records of odonates. We wanted to identify undersampled regions and habitats. We wanted to identify any rare and declining species. And then we also wanted to create a standardized protocol that could be used in the field so that we could kind of create a way to collect more data that's standardized throughout the entire state. And so to start, to kind of gather some data so that we could assemble these historical records, we collected data from insect collections in Illinois, a lot of Illinois natural history museums. We collected data from them. We also got some other museum data, I think the Smithsonian, and a few other throughout the country. We then also compiled data from verified naturalist databases such as iNaturalist and Odonata Central. And so by doing this, we are able to kind of understand the representation of various odonate species throughout Illinois. And this is important because a lot of museum collections tend to have older specimens. Some of the more recent Odonata Central and iNaturalist have more recent, and so this allows us to look at the populations throughout different time periods in the state. And so records from the museums and the naturalist databases accumulated in a total of 45,365 valid records of odonates in the state of Illinois. And so these valid records include ones, with the full date, the full species name, and GPS coordinates. We weren't able to keep any records that didn't have GPS coordinates, which was a lot from the museums because those were pretty old. But we needed those GPS coordinates in order to kind of understand the distribution of the species at that time period. And so using those 45,365 records, we visualized those locations of the records to identify any undersampled areas, which was our second objective. And so the first map on the left contains all the data that we collected, all of those records, which shows a pretty well covered state, specifically in the Chicagoland area and a little bit in the southern. And so that's all of those records. But our second map on the right is just data collected from 2011 and 2021. And we wanted more of an idea of kind of what these distributions looked like of the species in the last 10 years before we started this process. And we wanted something to be a little bit more current and relevant. And so here we kind of see that, Chicagoland area still is filled with a decent amount of records. But this kind of helped us decide to focus our specific surveys that we did in Southern Illinois, Central Illinois, and kind of that North Central Illinois area. And so our third step after doing that was to go through the records for specific species that were already on an Illinois watch list that was established by the state. And so there was about 27 species that were on this list. And it was important for us to kind of go through and look at the records from this database and kind of see if we can use that and the information that we have to decide what the conservation assessment might be for those species. And so one of those species that we did this for was the dusky dancer. And so the dusky dancer is pretty widespread throughout its range, even including outside of Illinois. It's not super common in Illinois as far as we know. It's a specialist. It's often found associated with streams and rivers, and with rocky substrates. They're slender, they're dark, they have limited blue coloration. The male of the species really stands out because it's one of the only damsels with almost an entirely dark abdomen. And also it has very limited blue on the abdomen tip. But you can usually find them perching on rocks or vegetation next to the water. And so using those records from the database, we created maps for historic, contemporary, and current records for each species of concern. And assessments are based on kind of presence-only data, and so we can't necessarily use them to help us with detection or occupancy, to kind of make those clear distributions, but it can kind of just help us lay out where they were previously found and are they still being found in similar areas throughout the years. And so for the Dusky Dancer, we had records from 1980 to 2010 that were found in Pope and Hardin County. And it actually comes from two streams, Big Creek and Big Grand Pierre Creek, which I found really interesting. And why I wanted to bring it up is because we actually, when we did our targeted surveys, we also sampled in Big Creek. And in 2023, we actually found a mating pair of dusky dancers in that spot. And so that was also in Southern Illinois. And then in 2024, we sampled on the western side of Southern Illinois in Union County. And we found two adult males at the Trail of Tears State Park in Illinois. And so that was really exciting, especially to find, you find a lot of common species when you do these surveys, but to find a species that is missing a lot of data was really exciting. And it was exciting to kind of think about the fact that our field surveys might have exposed a few new populations, and that would encourage us to want to do more targeted surveys. And so by looking at these maps and seeing kind of what they're showing us, our main suggestion for conservation would be to conduct future targeted surveys, and that the species is in need of conservation. Unfortunately, we did this process for all 27 watch list species of odonates, and about 20 of them had the same kind of predictions where they needed future targeted surveys and would be in need of conservation, which was a good portion of them. And so to kind of look at another species that maybe isn't as much as need in conservation was the elusive clubtail. It's a large, dark clubtail found throughout the Great Lakes region, and it's in large, non-flowing water bodies and large, sluggish streams. So the species is pretty hard to spot, and its name is rather justified. So that makes it kind of difficult to study. But when we look at our numbers for the species, we actually assembled about 153 records from our databases in Illinois. And based off of kind of what we see and the numbers that we see, it seems fairly stable. The abundance of records has kind of stayed about the same, which, the numbers may not look as big as we want them, but they are at least relatively similar across. And so, obviously the species tends to be doing the best in the northern half of Illinois, with a burst of reconfirmed localities in recent years. And, in northern Illinois, that's where the Great Lakes are. So it makes sense that maybe they're doing the best up in that area. Another cool fact with that maybe why we might be seeing more in that Chicagoland area, that's an area that's really focused for people that use iNaturalist and Odonata Central. And so there's a bigger group of community scientists in that area now, and there's a good chance that they're able to observe and record more than what we've seen before. And so overall for this species, this geographic distribution and kind of the abundance of the records, we would suggest that the species could be considered stable. We wouldn't necessarily call it like, protected or anything like that, but it's looking okay and based off of these maps specifically. And so most of you have probably seen the common green darner. around. It's not on our Illinois watch list, but I wanted to kind of take a common species and show you what their map looks like compared to these more rare or declining species that we have. And so, the common green darner, it's one of our most common and abundant species throughout North America. It's a large darner, often seen feeding over open areas. It is one of the best studied species of dragonflies out there. It's one of the big things that people study is their migration. They have a similar multi-generational migration to monarch butterflies. Um, and we tend to notice their migration mostly in the fall when they form giant swarms in evening hours. Similar to birds, they can actually be seen on radars when they migrate in their giant groups, which is really cool. And again, that tends to happen in September or October. And so here's kind of a comparison map for these species. So we know the common green darner is relatively abundant, it's stable, and you can kind of see the numbers. We have 220 records from pre-1980, we have 1980 to 2010, we have 63, and then in 2010 to present, we have 1,490. So the records are much more common, it's much more easier to spot, they're much more widespread throughout the state, and they continue to be prevalent in the areas that we would expect them to be prevalent in. And so it's kind of interesting to think about, is this because their populations are continuing to improve? Is it because we have really good community scientists? Is it because these species can adapt better to urban areas? And are we just seeing them more where other people are? And so it's kind of gives you some things to think about. Sadly, we can't answer those questions directly, but it's good to know that we are able to see data of species that are still doing okay and their populations seem relatively stable. And so with all that information, we then went out and did boots on the ground research. So we wanted to, target those specific areas that we felt were kind of undersampled, and wanted to collect data from those spots. So we avoided the Chicagoland area, but we decided to go to compile this data specifically. We wanted to create a standardized protocol for people to kind of collect data specifically on odonates. And so I'm going to kind of walk through some of what the protocol is that we did. And so for our sampling, it took place during peak flight season, which would be April to October for the majority of species. We would have 2 researchers at about 5 to 6 sites per regions in a combination of wetland and stream habitats. We wanted to make sure we were getting both wetlands and streams. We had 3 closed sampling rounds. They lasted 4 weeks, so we visited each region per week so that we had Southern Illinois, Central, North, Central, 2 Central, and then 1 Northern Illinois. And so we visited each spot each week. And within that week, we would go to 5 to 6 sites, but we would visit them 3 times in a week. So they were really long, kind of cyclical days of going back and revisiting sites. And we wanted to do that to make sure we were getting the best picture of a site we could. One visit to a site is just not going to get you enough data. And so by visiting 3 times in 1 week, we were kind of able to get a really good picture of what you can find at a site during that time of the year. And so over the 2023 and 2024 field seasons, we sampled at 46 sites total, a combination of both private and public lands. And we sampled in areas like Shawnee National Forest, Grafton, Champaign County Forest Preserves, Rockford, Peoria. We used actually quite a bit of RiverWatch sites for our stream sites, which was really helpful and great to kind of have those references. And so we did a lot of different types of sites, which was great. And so kind of our next step was to, we want to create these field methods that can then be replicated by other stakeholders and researchers. And so what was important is to make sure to collect all the data that you can at each site. And in order to kind of know what species are going to be occupying a stream or pond, you need to have that standardized method of even collecting the environmental data, not just the odonates. And so we kind of before we would collect any specimens, we would take a variety of data that included the habitat and environmental data. So, air, water temperature, invasive plant species, wind speed. So all of that stuff was done at every site we went to and every visit. Unless it was actively storming, we were trying to sample in all the weathers of other types in order to account for any species that may prefer conditions that are other than what would typical for odonates. I know most species like to be out in the open when it's warm and sunny, but there is a handful of species that would be found on colder, shadier days. And it was good to make sure that we were kind of accounting for those variables in case we could then predict something for that species. And so our equipment that we used for our methods, our most important equipment was obviously our nets. We needed butterfly nets to catch the adult odonates. However, you have to be a bit more aggressive when catching dragonflies than compared to catching butterflies. So we used a lot of butterfly nets because we broke them often. They were not as sturdy as we were hoping. Unlike the D-frame dip nets, similar to what you guys use in a RiverWatch, was what we used for the aquatic surveys. And these were much sturdier and they really helped us to scrape along rocks, logs, and streams and any of those microhabitats that we might find a nymph. And so kind of the first survey that I'm going to talk about is our aquatic sampling, which I'm sure most of you are familiar with. So for our protocol specifically, we had 2 researchers out in the field and we sampled along a 50-meter transect. Each researcher had their own 25 meters of that transect, and that's where we would look for dragonfly and damselfly nymphs specifically. And so for streams, we just ran a transect through the stream bed. In ponds, because it can be a bit easier to get stuck in the muck. We didn't really want to go into the pond as much. We kind of did a circular transect along the shoreline and kind of stepped in as far as we could. And then so this transect just kind of make sure that we were sampling the same amount of time and the same amount of habitat at each site. This kind of kept us within certain constraints. And so then within that transect, we would spend about 15 to 20 minutes sampling from the best microhabitats within either a stream or a pond. And we were really focusing, especially in streams where there's so many more opportunities for microhabitats such as vegetation roots, riffles, rocks, fallen trees, all things like that. We wanted to make sure that if anyone was burrowing or hiding, we really were able to kind of look in all spots. And so by kind of visiting these different microhabitats, we kind of knew we would get a larger diversity of species since they tend to use different microhabitats. And so that was kind of what we did for streams and ponds. It was mostly just kind of that bottom of the pond that we were sampling for. And so when you dip net, as you know, you probably pull up a lot of other things, sediment, rocks, mud, leaves, things like that, lots of other insects and organisms. And so we would have to sort through those to kind of make sure we were looking specifically for dragonflies and damselflies. Anything that we didn't need was just put back into the water. Luckily, dragonflies are relatively distinguishable because of how chunky they are. But damselflies were much more discreet and often looked like mayflies, and so it was important to take more time and actually make sure you're getting all the damselflies. We did find some other really cool things in the nets. We pulled up a lot of crayfish, and we also pulled up a tiny soft-shelled turtle once, which was really fun. And so for aerial sampling, which was our next part of the survey, I wish it was easier. It was not as idyllic as I had expected when you try to catch butterflies. We did relatively the same thing as you would to catch butterflies. It's just much more intense. They have that zigzag pattern that adults do when they're trying to escape predators. Well, they're really good at using that on humans as well. It's really effective. And so we chased a lot of adults kind of through streams and muck boots and kind of just trying not to fall on our faces. So aerial sampling was difficult to say the least, but it was really rewarding when you're able to capture a few species. And so following that same 50-meter transect that we did in the aquatic sampling, We then walked those same areas again. We focused on perching locations and sunny spots and sticks, basically anywhere that we think they might land, because that's going to be the easiest time to catch them. Sadly, not all species like to perch and land for you. A lot like to stay flying, like the darners, and those are really active. And so you spend a lot of time also trying to catch them in the air over your head. And those were really rewarding when you're actually able to do that, which didn't happen very often in catching one of those species. And so to wrap that whole survey up, after doing aquatic and aerial, I then would do a visual survey of the adults on the wing within that same transect. And I did this to kind of make sure that, when you're doing an aerial survey, you're spending so much time making sure you're catching things that you definitely miss kind of other species that might be flying around you while you're in the process of taking out another species from a net. And so I kind of wanted to just make sure I had time to just sit and watch while we're not like running through a stream or a pond, disturbing them. And so you could use scope, a spotting scope. I like to use binoculars to kind of get a good ID in the field of them. And it works for most of the species, which was really helpful. And so that added to our list of any observed species at a site. And so once we were done, we collect our voucher specimens. For adults, we really only try to take one individual representative for each species so that we're not over collecting from a population. And it does get easier to identify the adults in the hand, so you can just release them, which is great. However, for nymphs, it's harder to ID, and you need to bring them back to the lab to look under the microscope. And so we do our best to make sure we're not over collecting if the species start to look kind of similar, but anything that might look different, we try to bring back so that we can take a closer look and preserve these species under the microscope. And so in the lab, we try to break down the closest ID that we can using a bunch of various resources, dichotomous keys, lots of different books, things like that. And we focus on those observable traits that I had mentioned earlier. And so we're going to start at the largest taxonomic unit. So for us, that would be a suborder. Is it a dragonfly? Is it a damselfly? And then from there, we kind of move through to the families, genus, and then hopefully to a species. Based on the preservation or even the size of the specimen, we might not always be able to make it down to species, but we would try to get as low as classification as we could. We just wanted to make sure that our data was represented as best as it was. Those are just some like quick summary results. So we recorded 520 individual adults throughout the 2 years. We also collected 2,568 individual larval odonates, which meant I spent 2 years identifying 2,568 individual larval odonates. So that was a long, tough number, and I wish we didn't have as much, but it was great. It was a great amount of data. And then one kind of cool thing, there's about 154 species that are listed on our Illinois state checklist. And from that list, we found about 68 unique species. We do think that this number is probably an underestimation because unfortunately, unlike dragonflies have a great nymph identification guide where you can ID them to species. Damselflies don't have that, and so we can usually only get to genus if we're lucky for damselflies. But the hopes is that something like that will come out, and then we can add to that list, and hopefully that will increase our numbers. So another kind of exciting find from our field surveys was that we collected a nymph that was the interior least clubtail. And so this species is listed to not occur in the state of Illinois. And we actually found it in a stream in southwestern Illinois 3 times, 3 different times. And so the interior least clubtail is a small, slender dragonfly. It prefers clear, rocky, gravelly riffles or spring-fed streams. This species is often found, like their normal range is in the Ozarks. And considering since I found it just on the edge of Illinois on the southern western portion on the border between it and Missouri, there's a good chance, it's not that far from its normal range. That could just be a vagrant population utilizing that river. But this was exciting because, after looking at 2,000 plus specimens, I found something, that I hadn't seen before. And I used the really cool nymph guide that we have and in that left corner, you can see that's an antenna. And these are some pictures that I took from our microscope, and it looked so different and so unique that I know it may not look as unique to everyone, but it was just really exciting because you're like, this is different, and I couldn't find it anywhere on our Illinois list, and so that was an exciting find for us. And so another idea that we wanted to kind of give was just to help stakeholders and researchers to know when the best time to go out in the field is. And so in order to do that, we ran detection probability models for 28 species. And we picked those species because these species were found both in wetlands and streams and had a really good amount of data behind them, so that we could model kind of correctly the amount that we want. And so this helped us understand the likelihood a species is observed when present. Um, and this is also going to help us understand what environmental factors are important to a species and what might influence the probability of actually detecting the species. And so this will allow us to actually suggest to people, if you want to go look for X species, you want to make sure that the temperature is at least 25 degrees Celsius or things like that. The relative humidity is at a certain point. And so this, our variable list is not all inclusive. There could definitely be other factors that were really important for detecting certain species, but this is just kind of the general factors that we thought might influence a majority of species. And so based off of our recommendations, they've all been reported to the IDNR, but we found that from the detection probability analysis that we suggest prioritizing sampling during warmer temperatures and in bright conditions, and that's going to reach your higher detection probabilities for most of these species. And that's pretty common sense. We weren't too surprised by this. There's a chance that sampling on cooler and cloudy days may result in an underestimation of species because they're less likely to be active. But we also realize that, if you're specifically looking for a rare species who is still missing a lot of data, it would be recommended to kind of increase that search effort across different environmental conditions. And that way you can really help to kind of piece out which factors will influence their presence. And for example, we have a species like a mocha emerald that, tends to be in shadier streams and woodland streams. And you don't want to eliminate, those cloudy days because you might find species like that. And it's important to think about that as well. Another one of our suggestions is to make sure that you're surveying both wetlands and streams when you evaluate the local odonate diversity. And that's really important because, as I talked about earlier, a lot of species have so many different life histories that they could be overlapping in either wetlands or streams, or they could just be in one of them. And so it's common for most studies to occur at open wetlands because that's usually where adults are better observed. There's lots of sun, there's less trees and vegetation for them to land on. But however, we kind of found that streams provide really good foraging habitat for adults, but then they also provide great hiding spots for aquatic nymphs. And you're going to find way more diversity if you're going to have a bunch of different microhabitats to survey for. And we also kind of looked at our data set and we noted that the number of species that we observed, so individuals at a stream and a wetland were almost identical. So we found 47 like unique species at a stream, and then we found 49 unique species at a wetland. And so there is some overlap there. But the fact that they were so close is kind of showing how important and how useful both habitats can be, and that streams should not be, kind of pushed aside just because they can be cloudy or shady. They're also just a great spot to find nymphs. And so lastly, another goal was to decide on the best surveying methods. So obviously, while intensive, combining the aerial, aquatic, and visual surveys, we seem to contribute the best to the overall species observed. You get to target both the adults and the larval odonates, and that was really highly effective at contributing to the overall number of species that we found. And it's really great because nymphs can be in the water a lot longer than adults. Adults, have a dying off period. Nymphs have a tendency to last longer, so you can kind of find them throughout the season. That gives you more chance to add to your numbers. Aquatic surveys are also just really important because if you're finding nymphs, it's most likely that you're at a breeding site, and it's really important to know what a breeding site is, and it's important to make sure that you're conserving, that habitat as well, and making sure that's well known can be another part of that conservation plan. One of the downsides, though, obviously, of aquatic sampling is kind of how lethal it can be due to how often you have to view species under a dissecting scope. Um, and so that's something else to think about, the give and take of that. We also recommend that if survey time is limited, visual surveys could be probably one of the most beneficials than conducting just an aerial survey. When you're trying to run around and catch a species in a net, it's hard to make sure you know what's going on around you. So if you had to pick between either a visual or an aerial, a visual survey would probably be the best to use looking through binoculars. So that way you're not spending too much time trying to capture dragonflies that are flying overhead. But obviously, if time does allow, we would recommend both aerial and visual surveys because it really helped increase our detection rates and increase the overall species observed, which was really great for the kind of data we wanted to collect. And so all of our results and all of our databases that we created from this project have been reported and kind of given to our partners at the Illinois Department of Natural Resources. And all of that contains our suggestions for the odonate conservation and management plans. If we're given the opportunity to continue studying odonates in Illinois, we would want to continue our protocol to collect more samples. That way we can kind of increase those numbers that we have for some of those more rare species. And with better data and more data, we can make better predictions for detection and occupancy for other species. And it would also be really important to just focus more on visiting specific habitats that we know of more rare species like to inhabit. We did very general kind of locations, just kind of wherever we could find streams and wetlands. I think if we know a species tends to prefer seepages or certain types of wetlands, things like that, we would try to focus more surveys and more abundant surveys on those types of habitats, and that might help target some of our more rare and declining species. And for the majority of our species, we do think targeted surveys would be a really good, useful tool to kind of help take this project and take this information to the next level of conservation needs. And yeah, so with that, I just want to name a few acknowledgements. I had some really great collaborators, John Crawford, Ethan Kessler, Danielle Hake, and Michael Dresslick. Thank you to the Illinois Department of Natural Resources and the U.S. Fish and Wildlife Service for funding sources. And thanks to all my field crews that helped me out at NGRAC. And with that, I can take any questions. And thank you all so much for listening.
Thank you so much, Amy. And yeah, if anybody has questions, you can either put it in the chat or you can unmute yourself and go ahead and ask out loud. I think you just gave us so much information that we're all trying to process it.
Sorry about
Um,
that.
oh, no, it's wonderful. All right. Oh, here's a question. Are there any eDNA studies conducted?
Yeah, so I don't know the exhaustive list of them. We haven't performed any, but actually one of the big species that they do eDNA studies are for the Heinz emerald dragonfly. They do that up in Wisconsin is one of the areas where they know that there's a distinct population, and so they're testing out their methods specifically for that species up in that area which would be super useful because then we could potentially use that down in Illinois and see if we can find any more populations of the Heinz emerald. So yeah, that's a great question. I hope we were able to do more of that.
Great. And then we've got a question. Have you seen any declines in specific species during your study time?
So I haven't specifically, I think it's kind of hard to look at declines just over so we did two field seasons over just 2 years. I didn't necessarily notice anything different. I definitely really picked up on, when certain species are really abundant versus when they're not. But that would be another great reason to continue to do this type of protocols because I think with up to five years or 7 years, we might be able to get a better look at who's in real time declining.
Okay. Had a question of what guide are you using for dragonfly identification for nymphs? Garrison, Tennyson, thank you for the presentation.
Yeah, so my favorite is Tennyson. Some of those images that I had in my presentation were from that. I think that's probably the best dragonfly nymph one that I have found, and I found it really useful.
Okay. Are there any other migrating mates that appear on radar besides the green darner?
I believe the wandering glider will also migrate. I don't think they migrate in as large of groups as the common green darner. So I think the fact that the common greens swarm is really what helps them, and you can really see them on the radars migrating. So I think they're kind of one of the few. It's any of the ones. I have a feeling that other species will migrate with them, and so there's probably a mixed population, but it's mostly going to be the common green darners that are in those groups.
Alright. So from a conservation perspective, is there anything that you see as kind of the greatest threat to odonates in Illinois?
Yeah, honestly, one of the biggest things I've noticed in 2023, we had a pretty big drought. And I think there's the extreme weather habits is not going to do well for them. By the time I went out for some sites, so I would visit a site in early spring, summer, and then in the fall. I was able to collect all the data I needed in the spring, but then by the time I got out there, maybe even in May or June, some of those sites were dried up already, and I couldn't do any aquatic surveys. And it happened at quite a few sites, and that kind of didn't allow us to use that data, unfortunately. We just didn't have the same aquatic information as other sites. And so I think the extreme temperatures are really going to cause some issues. Odonates are really hardy and are really good at surviving things, but not having water is going to be pretty difficult on a lot of nymph and eggs stages.
Um, Thank you for the information. Were there any studies in the Cache River area?
We were just outside of the Cache River area. We stayed mostly in Shawnee National Forest when we were down in Southern Illinois, and then we went to some state parks when we were there. Certain areas are harder to get permits for, and so just based off of kind of what we had access to is kind of where we directed those site visits.
Right. And then, are you aware of the work done with Illinois Odonate Survey?
I definitely looked into it when I first started this project. I noticed that a lot of the work was up in Northern Illinois, and it's kind of hard when we're down here, closer to Southern. I did see that they were maybe starting to do some of that down in Southern Illinois. So I don't have any updates since I've looked at that a few years ago. So I would love to learn more about it.
Okay, any other questions? Made it through the list there. All right. Oh, Patricia says contact us about iOS.
Okay, will
All right.
do.
Okay. Well, thank you so much, Amy, for the great presentation, and thanks to all of our people who signed on for joining us tonight. Before you go, I do want to mention that our next Winter Speaker Series event will be Tuesday, February 24th at 7 p.m. Allison Brakefield of St. Louis University will be presenting her research on microplastics and freshwater mussels. You can register for this event through our Facebook page or by following the link in our most recent newsletter. Again, thank you very much for being here. Oh, and we did get another question. Can we get your results? So is there a report that's publicly available for this?
So right now the report is not publicly available. It is in the IDNR. Over the next sometime, we'll be hopefully getting these out in peer-reviewed publications. But the initial results for the report are not publicly available at this time. Illinois, the IDNR might make them available soon, though.
So yeah, it probably, I imagine that if it becomes publicly available, it'll be found somewhere on the National Great Rivers website. So keep an eye out for that. All right. Well, thank you all again and have a great evening.